Good morning, good afternoon, good evening, everyone. I'm Zumra, and I'm really excited to be here today. In this talk, I will share strategies for extending whole genome transplantation to new bacterial species. Specifically, I will discuss how we can disable recipient genomes and then revive the cells by introducing the synthetic genome, what we call zombie cells. First, I would like to start by introducing the current process of whole genome transplantation. Essentially, how JCVI-SYN1 minimal cell and its derivatives were created. The image you can see on the right gives you one of our key approaches, using yeast artificial chromosome to assemble synthetic genomes and then transplanting the assembled genome into a recipient cell. After that, I will go over some of the main challenges we have faced to move forward. Finally, I will introduce the concept of zombie cells. Whole genome transplantation allows us to turn a DNA sequence into a living, functioning cell. The process starts with a natural genome sequence. We redesign it, clone and assemble it in yeast, and transplant it into a recipient cell. This single step boots up the genome. Mycoplasmas were the first choice for developing whole genome transplantation because of their small genomes, simplicity, and lack of a cell wall, which makes transplantation easier. Yeast serves as an intermediate host for genome assembly and modification, and from there, the finished genome can be transplanted in one installation to create a living cell with synthetic genome. Let's look at the stages of this one-step genome installation. For the genome, we grow yeast carrying the genome, capture the cells in agarose gels or agarose plugs, and lyse them for the intact genome. We then prepare it with enzymatic digestion. For the recipient cells, calcium chloride neutralizes negative charges.
Yourself, please? Thanks.
Thank you. Polyethylene glycol promotes membrane fusion. Our synthetic genomes carries a tetracycline resistance marker, so only transplanted cells survive on tetracycline plates. Within about 3 days, blue colonies form due to the LAC-Z gene, showing successful transplantation. In this case, only synthetic genome-containing cells will survive. Genome transplantation is powerful, but it is demanding. The experiment is fragile. One mistake can cause failure. It takes skill and sometimes a bit of luck. Because of these challenges, researchers have looked for alternatives to genome transplantation. George Church's lab and Jason Chin's lab work on synthetic E. coli by iterative replacement of 50 to 100 kilobase pair synthetic genome in native E. coli. Here's one example, just came 2 months ago from Jason Chin's group. They engineered an E. coli, one second. They engineered an E. coli with a reduced genetic code, only 57 codons instead of the usual 64. By the iterative replacing 7 codons across the 4 million base pair genome. If whole genome transplantation was broadly available, such a recoded genome could be built in weeks rather than months. Why don't we have whole genome transplantation for other bacteria? Originally, Mycoplasma genitalium was chosen for whole genome transplantation process because it has one of the smallest genomes known, just 580 kilobase pairs. For years, our group tried transplanting genitalium genomes into other genitalium strains, and every attempt failed. Then, transplantation of mycoides into capriculum came as a big success. Naturally, we thought we could repeat that with other species, but when scientists tried interspecies transplantation in E. coli plasma and Haemophilus influenza, they ran into the same dead ends as before. At this point, we had to ask ourselves, were we incredibly smart or just incredibly lucky to stumble on the one species pairing that worked? The odds that we have already found the perfect donor-recipient combination for genome transplantation are pretty low. Even though, right? quickly. Even though this technology has been around for 15 years, we can turn genome transplantation's failures into lessons for expanding recipient species. Even if we can clone a genome in yeast, it cannot be used unless it's successfully transplanted into a living cell. Yeast is just a safe place to hold the genome, but to function, it has to go into a recipient cell. Although many bacterial genomes have been cloned in yeast, only microbes clade have been successfully transplanted. This work, conducted by Carol, a former member of Glass Lab, and represents her current research. Her findings indicate that the success rate of the transplantation decreases significantly as the phylogenic distance between the donor genomes and recipient cells increases. This decline in transplantation efficiency in this study shows us universal recipient cell ID is not going to work. The solution will likely come from focusing on intraspecies transplantation. Also, all of these successful transplantations were into Mycoplasma capriculum recipient cells. We definitely need additional recipient cells to move forward. Unlike nearly all other bacteria, capriculum recipient cells do not engage in homologous recombination. We can use antibiotic-resistant markers to select for transplanted cells. However, this approach can result in false positives through homologous recombination in other bacteria. We attempted an intraspecies transplantation using genitalium. In our initial efforts, employing a single selection marker led to homologous recombination within the recipient cells. In order to avoid this issue, we designed the donor genome to include two antibiotic-resistant markers located far apart from each other. The transplantation reaction was plated on agar plates containing both tetracycline and kanamycin. After 6 weeks, we observed 2 colonies. However, sequence analysis revealed that these were false positives, resulting from the recombination of a segment from donor genome, specifically the region containing the antibiotic-resistant markers, into the recipient cell genome. In order to overcome homologous recombination, we can inactivate the recipient cell genome prior to transplantation so that only cells with the new genome will survive. This is the basis of zombie cell concept. After killing the recipient cells, we revive the cell with a synthetic genome. Mitomycin C is a chemotherapy drug and known with its DNA cross-linking properties. It specifically targets a guanine nucleoside within the 5' CpG 3' after reductive activation followed by 2-nitrogen alkylation. The covalent bind between G and mitomycin C, you can see here, disables the genome. When capriculum is incubated with mitomycin C, the majority of the cells die. However, we can still recover for transplanted cells. Live cell count and transplantation count as a function of mitomycin C concentration shows dramatical drop in the live cell count. Even though the cell count lost about 1 millionfold, our transplantation efficiency only decreased about 10-fold. This raises a fascinating question. Are we transplanting into dead cells or alive cells? No transplantation is observed above 20 microgram per milliliter mitomycin C concentration. This is little dosage for the cells that even their RNase inhibits. Overall, we are aiming to use mitomycin C concentration in DNA inhibition dosage. Let's take a closer look at the data from the previous slides with a statistical analysis. When we do the original genome transplantation with about 10 billion recipient cells, we only get 68 transplants. I know it's very low yield process. That roughly means one transplant can be achieved from a few 100 million cells. When we treat the recipient cells with mitomycin C, the efficiency suddenly skyrockets. With only 7,000 alive cells, we get 25 transplants. That's equivalent to 300 alive cells per one transplant. This means roughly 500,000-fold increase in efficiency. So are we transplanting are we making super transplanters with the mitomycin C treated cells? Or is it more likely that we are transplanting into dead cells, which these dead cells can no longer replicate their own genome, but they are able to host the donor genome? We don't think we are making super transplanter with a few living cells. When we took mitomycin C survived recipient cells, grow, and perform transplantation with them, the efficiency didn't show a big difference. The first genome transplantation experiments were originally developed on the foundation of chemical transformation assays. If mitomycin C creates super transplanters, they should also behave as super transformers. Hence, we directly compared transformation and transplantation under identical mitomycin C conditions. Because transformation requires functional replication, it should only succeed in viable recipients. Here we treated the recipient cells for 2 hours with 2.5 and 5 microgram per milliliter mitomycin C and 1 hour with 10 microgram per milliliter mitomycin C. We see that after mitomycin C treatment, transformation is completely lost, meaning a few living recipient cells are definitely not super transformers. Despite no transformation, we can still see a successful genome transplantation. That means transplantation is occurring into the effectively dead in terms of original genome activity cells. Here we see the original genome transplantation process. Brief mitomycin treatments cross-links the DNA of the recipient cell, but leaves its transcription and translation machinery intact. This creates a short window where the donor genome can be introduced and expressed. Mitomycin C essentially shuts down the recipient genome. Before transplantation, recipient genome cannot interfere with donor genome anymore. Afterward, transplants are plated on non-selective media because only transplanted cells revive. Overall, our motivation is to expand genome transplantation for other bacterial species. The zombie cell concept is a promising approach to overcome the problem of homologous recombination. And next year, I'm hoping to show you E. coli recipient cell species that we will create with mitomycin C treatment, or the problems that we will face besides homologous recombination. Most of the experiments were carried out by Nasira, Vanya, and Filan. Now I'm focusing on writing and wrapping up the last experiments. And right now we are putting together the zombie cell paper. Also, we are working on whole genome transplantation protocol paper, which everyone can utilize with the detailed steps and videos of them. I'm so happy to be a part of this project. And now I would like to thank everyone for listening, and I would like to extend my thanks to John for his invaluable mentorship so far in this journey. I'm also grateful to my colleagues in the Glass and Suzuki labs for their support. A special thanks to Elizabeth, Jane, and James for their contributions and encouragement in our weekly meetings. This is just the beginning, and I'm truly thrilled with our results so far, and I'm looking forward to your questions.
Very nice, Zumra. Questions?
Can I ask a question?
Please,
Hi.
Bogomil.
So that's beautiful. This is just so fascinating. Zombie cells definitely great thing. Now, so you talk about this homologous recombination, but is that really, but you're using species that have no homologous recombination. So are you proving the point that this is actually helping with that aspect,
This is such a great question. So far, so this came up from the genitalium species, since we have seen the homologous recombinations happening there, and we thought this would be a great thing because most of the bacteria are having the homologous recombination. And now I would like to move forward with E. coli, which has a homologous recombination as well. So this is helpful for them. But I'm not sure if it is good for a coloplasma too.
Yeah, so because I keep also thinking, because you can go, from example, from capricolum to capricolum or mycoides to mycoides. Now, you're talking that there are more cells. Obviously, you're getting more transplant per cell when you treat, but overall, do you get actually more transplants in the reactions? Because in the end, so
No,
can you,
yeah, we actually don't get more transplants. If anything, I can tell you, like, we are getting half of the transplants. Like, generally, it is what we get untreated versus treated with mitomycin C. But the most important thing is it is going to go through like the elimination of the recipient's cell genome. So that's the whole promise of it.
right? And so, yeah, because obviously we're struggling with E. coli plasma, and we want to see if that would be better in the end. So is it better to actually take and delete the 1 or 2 recombination genes, or actually pre-treat that? But that's, again, super fascinating, and that you can actually do this. This is really, really amazing. And yeah,
Sure.
so good luck. I just, from practical reason, it's like, is it better to actually delete the recombination or pre-treat them like that?
I see. So in E. coli plasma, you guys don't have any homologous recombination proteins?
No, not really. We restore that, but we have strain that doesn't have it. So
Okay. So I guess it is better so not to use mitomycin C, so you can still have the live cells, I believe. But of course, like transplantation is very interesting and it's not very straightforward, and we don't know the mechanisms of it. So it's hard to say, to be honest.
Okay, I won't take time. Thank you.
Of course. Thank you for the question.